I play a very little role here. I oversee the sequencing facility, and it's a team of about 20 people, and we actually have quite immense capacity to process samples in parallel with 10,000 samples going through DNA extraction every day. We can run up to 10,000 concurrently through the amplification stage. So it gives us some really powerful capacity to support large-scale projects. And of course, we different from a lot of other facilities because we work with a lot of diverse organisms with different level of preservation and quality of DNA. And the protocols are optimized to be good enough to work in a wide range of different taxa with different quality of templates. So we have built quite a community, and we did serve researchers from more than 50 countries with 300 different research groups. And over the years of the iBOL and a decade of DNA barcoding, we have contributed at least 75% of all barcode data in the BOLT system. So it actually takes quite an effort to collect that material and then organize it into a format that is lab-friendly. And I put a slide here just to illustrate one point that from my personal experience, where a team of 3 people working for a week doing proper databasing, data accessioning, voucher imaging, can assemble, and tissue sampling, can assemble 20 plates. And then when it goes into the lab, the hungry machine eats it up in less than a day. So we'll focus on this mess over here. And it's not very bright, but in principle, it's a very simple straight line without any details on the underlying workflow. And the classic Sanger-based DNA barcoding all has the same stages with lysis DNA extraction, PCR sequencing. We use spray cleanup, and then it goes into the instrument. And if you're not doing 95 samples at a time and 96 well plates, then you're doing it for real. And of course, when you look into the cost, and like again discussed multiple times already, everybody's looking after $1 barcodes and less. You won't see dollar value in this presentation because my answer, it always depends. Like if you work with degraded template, if you want better quality of DNA, you will have to use an appropriate protocol that drives the cost down. But we still make efforts to make the process efficient and reduce the cost to allow more samples to be processed with limited resources. So here's one example where major contributors are consumable costs for DNA, for PCR stage and 96 well. About 50% of that is reagents and about 50% plastic labware. By moving into 384 well format, we actually reducing the total cost of the stage by about 20%, and then the primary component that was remaining there is still the laboratory consumables like the robotic tips, for example. So there's still plenty of opportunity to improve these processes, and Sanger sequencing definitely has its limitations. We can do a little bit more here. So we did build immense capacity. Bulk of the work that we've done through the years was linked to the iBOL phase 1. We have had some major changes in the protocols and introduction of the 384, adoption of the processes and 384 well plates. And then we built up to 1 million a year to close off the iBOL phase 1. And then what you see here, those are bars for the samples we extracted each quarter. In our best quarter towards the end of the project, we were basically working around the clock to hit that target. And then that red dashed line follows closely that graph, those actual samples that went through Sanger sequencing. But again, Sanger sequencing has high costs. You actually can deal with mixed templates, has some limitations on GC content, homopolymer attracts, and does need higher quantity of the input material. Yet it is actually the sequencing technologies is embedded into the barcode standard, and if you care about low cost, it actually offers you lower entry cost compared to NGS sequences, because just to press a button on the instrument sometimes can exceed $2,000 or $10,000 depending on the platform. So there was definitely a push to adopting high throughput sequencers, third, second generation platforms to reduce the sequencing cost. And there are different limitations on all the platforms, but they all can be used to generate barcode data. The differences are their compatibility with existing quality standards, scalability of the Amplicon library preparation, and then complexity of the data analysis. So we've settled on a particular platform, and I'm just showing the results from the recent paper that we published as a validation for this approach, where sequences that you generate from Sanger and smart sequencing are absolutely identical. And it's just a small subset of a tree with almost 10,000 samples. So I'm not going into comparison of all those experiments and the reports. It's all available in the paper, and I don't think I'll have the time. But with the adoption of smart sequencing, there is actually one big change. So as you already noticed from Jeremy's presentation, we did start declining in our throughput after the end of the iBOL because we were reevaluating our processes, but at the same time, about 80% of the material is coming from the institute, and 20% come from the external submitters. So what you see here is actually the red line deviates quite significantly from the bar graph. So this is the number of samples we extracted. This is how many of those extracted went through Sanger sequencing. Everything else that you see here, the Ontario Provincial Park project and then global malaise that we did this summer, went through the SQL platform. So how does it actually work? The targeted sequencing on SQL platform is, well, it's generally the way the smart sequencing works on any of their platform. It's a ligation of the hairpin adapters to the target amplicon that consists, yeah, it's very pale here, but then hairpin adapters, and then your insert, and then it creates a structure that is called the SmartBell template. And you have those 45 long adapters on each end, and then your amplicon, and then all of that, after a proper process, is loaded on the smart cell, and then the sequencing itself happens. So the smart cell has about 1 million microRLs, the zero-mode waveguides, where the enzyme with the template is immobilized close to the light source, and then the immobilized template has strand displacing capabilities, and it pulls the template when the new base incorporated, it emits fluorescent signal, and then it's recorded, and then you can have data collection up to 10 hours with one type of smart cells, or up to 20 hours as of this year to generate even longer reads. And then the 8 million version is still on the track for the release in 2019. So what happens is that your sequence generated from the SmartBell template, it represents concatenation of your molecule with forward reverse trend that are informatically collapsed into circular consensus sequence that results in high fidelity. The raw read error is 10 to 14%, but because the error is random, the consensus sequence becomes a high fidelity sequence, and the longer that polymerase read, the more accurate the consensus will be. So our target template that we favor is actually 811 bases, and this is what is, oh, it doesn't even have any colors anymore. So then this is your target region with primer sites, and PCR1, you get the primers that are tailed with forward and reverse PacBio adapters. This is a universal system that you can have any loci targeted primers with the same adapter, and then you can use the same set of barcode tags to work with plant, animal DNA. You can do multiple loci, but of course that complicates the reaction because you need to do a second PCR where you use that PCR1 product to index it with universal molecular identifiers, and by using 96 forward and 96 reverse unique molecular tags, we can get that combination of 9,216 unique amplicons in your run. So then this is what sort of it might look like when you try to do a heat map of your samples. It really washed out now, it's high intensity, but this particular heat map has 10,000 samples, and each intersection is a different combination of the forward and reverse barcode. So this is what the new workflow looks like when you simply replace Sanger stage with a SQL platform. And then you still go through DNA extraction, you do 384-well PCR, PCR2 stage to index your libraries. Then you pull everything together into a single tube, and then you run through the complicated, I'm not showing all details, but that's right now 2.5-day workflow to go to generate the library, and then 10, 20 hours, depending on what you want to do on the sequencer. But focusing so much on the cost of sequencing and the choice of the platform is somewhat misleading, because in this case, for this particular set, when we do 9,000 samples, 87% is actually all the work that needs to happen before the sample goes onto the sequencer. So this only little component over here that corresponds to sequencing itself. So you need to focus on improving the efficiency of the steps before you even start sequencing, and consider the implications for the quality of the final result. This is what the results of circular consensus will look like after you actually assemble those reads. Our target insert is about 811 base pairs, and then there's some distribution, but the majority of them fall into that category. There is a second peak here. Some of the groups have a deletion of about, I believe, 9 nucleotides, so then they fall into a shorter category. But there is a range of the polymerase reads, and then you see that for some of the samples, you only get 20 passes through the insert. So then you assemble consensus from the 20 subreads of your molecule, and in some cases you get as many as 40. So then most of the reads in this particular application are at least QV30 with 99.9 predicted accuracy. But what bothers me personally, until recently, was 1/3 of your reads end up, 1/3 of your total reads from a single chip end up as a useful CCS reads. Regardless, even with that output right now, we're getting, depending on the input material quality, more than 70 to 85% success without dealing with any ambiguous reads. So recent improvements of the PacBio chemistry and the smart cells are changing that pattern, and they found a way to shift the read distribution. So most of the reads now that are generated from the circular consensus sequences alone, thus resulting in most of those reads contributing to your final data. So we expect the success rate would be much higher. If we improve in the efficiency and we want to have a very specialized system, we can further drop down the complexity of the sample prep and make something to make Paul very happy, because all we care about is just barcode, the Malay strap samples at the absolutely minimum cost. So this is something that we're trying to optimize right now by generating your dual index amplicons in a single step. That means that you no longer have a universal system. You would have to have a separate set of tags for, say, if you want to do another loci, but because 80% of the samples that we do are Malay strap samples, I guess that's justifiable investment. So this is what the new workflow starts looking like. You lose another plate, and you are limited by the existing liquid handling infrastructure to achieve further reduction of the volumes, because 99% of this amplicon pool that we generate right now is ultimately not used for the sequencing library prep. So I find it very annoying that it's not as efficient as it should be. So the next thing would be to bring that reaction volume down even further, and I'm not permitted to say how we're gonna be doing it, but the future is sound. So this is my concluding slide where I'm trying to make the point that the pre-sequencing steps are very important in terms of overall cost and the quality of the barcode library, and you definitely need to have a high throughput facility that has automation capabilities to work at this scale, especially if you want to go after this ambitious target. So for us in the current situation with the current capabilities and the upcoming upgrades, PacBio SQL is definitely the platform of choice. This may change, but as long as we figure out how to move the samples from the field into the lab, into the sequencing stage, and then through the data analysis, I believe we already solved most of those. And some things that probably need to be considered in the discussion are the bioinformatic costs. They often neglected, but we had a serious experience ourselves, like what difference that can make, but then that has a real cost. The adjustment to the barcode standard, and then the endorsement certification for the analytical facilities. And of course, I wanted to see the faces. Like most of that work on the analytical protocols, especially automation, is because of Natalia Ivanova, lead DNA scientist, and then the rest of the team who is responsible for running the samples. I also extend my gratitude to my colleagues, and I think I'm out of time for questions.